Welcome to the cellular tour. Today we will be touring an animal cell. If you wanted to see a cell wall or chloroplast up close, then this isn't the tour for you. An animal cell does not have a rigid supportive structure, aka the cell wall, and it also does not have any chloroplast to synthesize energy for food. However, an animal cell has other unique features like lysosomes and centrioles, which you will see today. But how are we going to get into the cell? Through the cell membrane. The cell membrane is a thin, flexible barrier that surrounds the cell. It is made of a lipid bilayer. The lipid bilayer contains a phospholipid polar head and a nonpolar phospholipid tail. Inside the lipid bilayer are also molecules such as carbohydrates, cholesterol, and glycoproteins and glycolipids. Also in the cell membrane are peripheral proteins and integral proteins. The integral proteins go all the way through the membrane, while the peripheral proteins only stay on one side of the membrane. The cell membrane is semi-permeable, meaning it allows only certain materials to pass through, and certain materials cannot pass through. One form of transport is passive. Passive transport requires no energy from the cell. The molecules flow from high concentrations to low concentrations. The diffusion of water through the cell membrane is also passive transport. It is the process of water moving through a selectively permeable membrane and is called osmosis. However, we are too big of a group to get through the cell membrane through passive transport. We need the help of a transport protein to get through. We'll be going through the membrane by active transport. Active transport requires energy usage or the help of a protein. When molecules are too large, polar, or have a charge, active transport is necessary. There are subforms of active transport, but the main processes are endocytosis, which is when the cell takes in solid material, and exocytosis, where the cell expels material through the cell membrane. Now that we've successfully passed through the cell membrane, you can see that the cell is filled with a substance that is called cytoplasm, and it is made up of cytosol and water. It fills the entire region of the cell between the nucleus and the cell membrane. Throughout the cytoplasm, you can see fibers extending everywhere. These fibers make up the cytoskeleton. The cytoskeleton is made up of three different molecular structures: microtubules, microfilaments, and intermediate filaments. Each structure serves a different purpose, but overall the cytoskeleton supports the cell, produces motility, organizes cell structure and activities by anchoring organelles. You can also see microtubules in a certain arrangement that are paired together. These are centrioles, and they are unique to only animal cells. The centrioles help move and organize chromosomes during mitosis. Also, peppered around the cell and the cytoplasm are ribosomes. Ribosomes are sites for protein synthesis. This type of ribosome is known as a free ribosome because they are suspended within the cytosol. These free ribosomes produce proteins that are able to be used within the cell. Unlike a plant cell, the animal cell does not have a large central vacuole. The vacuole in an animal cell is used for storage. The vacuole stores food, water, waste, and other materials. Over there, the organelle with a sort of zigzag lightning bolt design is the mitochondrion. The mitochondria of the cell produce energy, or ATP, for the cell from sugar, fats, and other fuels. Think of this organelle as the powerhouse of the cell. Another organelle that you'll see in the cell close are lysosomes. Lysosomes are membrane sacs, but are made up of hydrolytic enzymes. These enzymes are used to digest food, old or broken cell parts, and other molecules. Now we are going to head towards the nucleus. This is what you can say is the brain of the cell. The nucleus contains the genes, or DNA, that control the cell. However, we cannot see the nucleus just yet because it is surrounded by a barrier that is called the nuclear envelope. The nuclear envelope acts as the boundary between the nuclear contents in the cytoplasm. The nuclear envelope is a double membrane that is perforated with pores. We are going to enter through one of the pores and view the nucleus and the nucleolus inside. Inside the nucleus there are many wiry-looking things. These are called chromatin. They are made up of DNA and proteins. Chromatin are unwound, decondensed, and dispersed DNA. DNA is always in this form when in use, unless the cell is preparing for mitosis. During mitosis or meiosis, the DNA is wrapped around proteins and is tightly coiled and condensed. Also inside the nucleus is the nucleolus. The nucleolus is where the components of ribosomes are synthesized and created. As we leave the nucleus through another pore, you will be able to see some of these proteins. Outside of the nucleus is the endoplasmic reticulum. There are two parts of the endoplasmic reticulum, the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough ER is nearest to the nucleus. This is because the rough ER transports the proteins made in the nucleolus to transport vesicles. You can see the ribosomes that are attached to the rough ER. These bound ribosomes synthesize and produce proteins that are used outside the cell or inside the cell membrane. The rough ER attaches carbohydrates to some of these proteins. To our right or above, you can see the smooth ER. There are no bound ribosomes on the smooth ER. The smooth ER does not synthesize proteins like the rough ER. Instead, it synthesizes lipids, phospholipids, and steroids. It also metabolizes carbohydrates, detoxifies drugs and poisons, and also stores calcium ions. Now that we are done with the endoplasmic reticulum, we are going to travel with one of the vesicles. We are going to go onto a transport vesicle and go to the Golgi apparatus. The transport vesicle carries products from the endoplasmic reticulum to the Golgi apparatus, which are flattened sacs. Inside the Golgi apparatus, the products from the ER are modified. But that's not all the Golgi apparatus does. The Golgi produces other molecules as well as lysosomes and secretory vesicles. Like the transport vesicle, the secretory vesicle moves things from one place to another in the cell. The secretory vesicle takes the Golgi modified products to the cell membrane. It fuses with the membrane and releases the materials outside of the cell. This is how we're going to exit the cell membrane. We're going to exocytose. However, before we leave, take a look to your left. These hair-like structures you see are called cilia. Cilia help cells move, and they also help move fluid or materials past the cell. We are now at the end of our tour. I hope you learned a lot about the animal cell. After all, you are made up of many of them. Really, a lot of them. In fact, it's cell theory. All living things are made of cells. Cells are also the basic structure and function of living things, and new cells are made from existing cells. Thank you for joining me today, and I hope you look into a plant cell if you have not already.